Hello, my name is Victoria Leeder and I am a senior at Eastern High School. The title of my project is Vitamin Enhanced Agglutination. The purpose of my project was to discover if by adding vitamins to antibodies I could increase their efficacy. I used salmonella antigens and antibodies for my project. I used 3 different types of vitamins, vitamin B12, vitamin C, and vitamin H, at 2 different concentrations of 10 and 100 milligrams per milliliter. My hypothesis was that vitamin C at 100 milligrams per milliliter would cause the greatest increase in efficacy of antibodies. I tested this hypothesis by varying dilutions of antiserum at 1 in 5, 10, 20, 40, 80, and 160 dilutions. I then put these into a well slide with the antigens, the salmonella antigens, and with my vitamin solutions. I then put these under a microscope after waiting for 10 minutes and checked the agglutination patterns. The highest dilution at which I saw agglutination present was the one that I recorded. I did 5 trials of each vitamin and each concentration. I then took the highest dilution denominator and I averaged these between the 5 trials. I put them into graphs, and as you can see here, I have 2 different graphs displaying the same information. This graph here displays average dilution denominator by vitamin. This is vitamin B12, vitamin C, vitamin H, and the control is in gray. Green is 10 milligrams per milliliter concentration, and blue is 100 milligrams per milliliter concentration. As you can see, vitamin C was the most effective between the vitamins and also between the control. In this graph here, you can see them organized by 10 milligrams per milliliter concentration and 100 milligrams per milliliter concentration. Vitamin B12 is in red, vitamin C is in orange, and vitamin H is in yellow. Once again, you can see that vitamin C caused the greatest increase in efficacy, especially at 100 milligrams per milliliter. In these pictures here, you can see positive controls of agglutination, and here you can see a negative control of agglutination. Agglutination is the clumping of antigens and antibodies. I was looking for these in the highest dilutions in order to gather my results. I then put my results into statistical analysis tests to determine their significance. I first chose to use a 2-way ANOVA with replication. I chose to use this because I had replication in my data and I knew that it would be the best way to test for significance. I found that I did in fact have significance because my p-value was 5.37 times 10 to the negative 8th. This showed significance and that my vitamins had caused it. However, I wanted to know precisely which vitamin at which concentration had caused the significance. So then I put my results into a 1-way ANOVA with a Tukey-Kramer test. The Tukey-Kramer test showed me that my significance was caused by vitamin C at both concentrations. However, the variance in concentration was not significant. This meant something because I had expected the change between 10 and 100 milligrams per milliliter to be slightly significant. However, it was not. But this did show that I could reject my null hypothesis that vitamins had no impact on antibody efficacy. So I was able to conclude that my hypothesis was correct, that vitamin C at 100 milligrams per milliliter did in fact increase the efficacy of antibodies, and that vitamins do have an impact on antibody effectiveness. This is important and can be used in everyday life because when people get sick, they want to know if vitamins actually do aid in their wellness and in their treatment, especially in a time such as now when there's a pandemic and there's lots of sickness going around. Knowing that vitamins actually do something to increase your well-being and to aid your body in fighting off disease is very important. Thank you for listening to my presentation.